Welcome, everyone, to the 38th annual meeting of the Hudson Hoagland Society, which is about discovery. And speaking of discovery, Alexander Graham Bell discovered the telephone. Why is Thoreau on a telephone? Because we've got some technical problems with my audio here at the house, and therefore you're going to be hearing me through this medium. These are the occasions when we thank you for your support, hear the Chancellor's update of institutional progress, and then a speaker who embodies both the passion and the art of biomedical science. And as for your support, if I could go to the next slide, I'm pleased to announce the recipients of the 2023 Worcester Foundation grants that your gifts make possible. These 5 proposals were chosen from 37 submitted, all of which were outstanding, but these were deemed especially so. 2 of them are as clinically proximal as any proposals we see. And I'm particularly pleased that Dr. Philip Tye's proposal was selected by the Bassick Family Foundation as their designated grant this year. Why do I say particularly pleased? 1, because it's such an annual pleasure for me to meet with the Bassick family and discuss the finalist proposals, for them to then make their selection. The second reason I'm particularly pleased is that the collaborating investigator with Dr. Tye will be Dr. Kevin Donahue of our cardiology division, a world leader of heart arrhythmias, especially AFib, and a former Bassick Family Worcester Foundation grantee himself. These grants and those that have preceded them over the years are the seeds of innovation that constitute the first step in the knowledge acquisition process that in the great majority of cases have led and will lead to expanded scale via federal funding, typically through the NIH. As I say every year, I cannot emphasize enough that you are the discovery capital catalysts of these catalytic scientists. And you are to be congratulated, and you are to be proud. It is a great thing you do. Thank you. It's my privilege now and pleasure to call upon Dr. Michael Collins, Chancellor of the UMass Chan Medical School and Senior Vice President for the Health Sciences for the University of Massachusetts. Dr. Collins, we look forward to your remarks and know that they will convey as much as ever the extraordinary momentum of the institution you so ably and proudly lead. Dr. Collins.
Thank you, Dr. Peterson, and good afternoon, everyone. And on behalf of Dean Flott and myself, let me add our appreciation to those of you who are on the meeting today for your continued generosity. It's just such a boost of confidence that folks like you are continuously supporting our institution. And I'm extremely grateful for your continued generosity. Now, Dr. John Harris has the meat of today's meeting, and so let me get through this relatively quickly so we can learn about these skin diseases. We're pretty proud of Dr. Harris. He's one of ours and is doing great things, so it's very exciting to know that he'll be talking shortly. So 50 years ago, this was the pasture. The building in the front corner right there where the dot is, that was the Shaw building. And that's where the first 16 or so medical students came in the fall of 1970. And you can see in the background that the medical school and hospital building had begun to be built. There was a power plant. The lake is to the front there. If you haven't read the book, The Boys in the Boat, you should read it. It's a terrific team building book, and it actually takes place on this lake, Americans training for the 1932 Olympics. And so it's pretty exciting. This was the campus. And if we go to the next slide, boom, 50 years later, here we are. And we'll be celebrating our 50th commencement in a few weeks. And that's pretty exciting. And, you know, as I think about it, I can't imagine that if folks were even blue skying what the institution might be 50 years later, that they would have had this sort of vision in their mind. And so we're pretty excited about that. I say institutions have their moments, and I really do believe this is our moment. Now, one of the things that's very important about our institution is that everything we've done to advance it has been the result of a strategic plan. We've done 3 now. They have 5-year durations. We update them every year to 18 months. And so when you see the Sherman Center, you see the VA building, or you hear about increase in class size, these are all the direct result of very thoughtful and intentional thinking on behalf of our academic community. And if you would like, you can go right on the website and piece through this. There's a lot of really great stuff. And I hear from my colleagues around the country that they love to look at this kind of stuff, so it gives them great ideas. And if you look at the mission statement, it's fairly clear. We're here to advance the health and wellness of our diverse communities throughout Massachusetts and across the world. Now, if you think about at the beginning, there was very much a Massachusetts focus, but we are a global leader now in medical education and research, and we're darn proud of that. Next slide, please. Now, in entities, if you could get everybody to be on the same page and say the same phrase, this would be the phrase I would like folks to say, that what we are about is to change the course of history of disease. And whether it's our clinical care, our research efforts, preparation of our learners, changing the course of history of disease is truly what we are all about. And in the next few slides, I'm going to show you why we're so excited and why I think this is a momentous time. Now, I'm sure that most of you know that Dr. Gerald Chan and his family, through the Morningside Foundation, made a very big statement in 2021 by providing a $175 million gift to help us to transform the campus. Now, what's extraordinary about this gift is that it is 100% unrestricted to the chancellor's discretion. And these are like dream gifts in higher education. There are the donors that will come in. I remember once there was a donor I was dealing with that wanted a Japanese tea garden on a campus. It wasn't ours. And, I was all for it. I had no issue with tea. But, once we build the thing, and then we got to buy tea every year, we got to get somebody in there to boil the water and serve it. It just wasn't something I was interested in. But when you get $175 million and it's completely unrestricted, that is really a special gift. And what it's done is to capture the attention of philanthropists across the globe. And I'm pretty excited about that. So these are the brand names that we now are using for our institution and the 3 schools. For those of you who don't know, Morningside is the name of the foundation. T.H. Chan is the Chan family's dad, and Tan Ching Fen is the Chan family's mother. Now, I would just say that if you name a school after your father and your mother, you have a long-term interest in it, and I really do believe that this is the intention of the Chans. I have the great privilege of talking to them almost every week, and it's really very, 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 very exciting. Okay, now let me just show you a couple things just to give you a sense of where we are. I'll kind of by the numbers. So we have 3 schools. The medical school right now admit in the class that came in the fall, we admitted 175 students. We're in the process of admitting 200 students. Next year, we're going to go to 225 students. And if all goes well, we'll be at 250 in the near and foreseeable future. Our graduate school of nursing is unique in that everyone in the nursing school already has an undergraduate degree. So you don't come to this is a graduate school of nursing. So, so let's say you went to Wellesley or Brandeis or Boston College and you studied music or French, and now you decide you want to go into nursing. Those are the folks that are in our nursing school. Very, very, very highly accomplished learners, by the way. And then in the graduate school, these are the folks who are getting PhDs and supporting our research enterprise. We have 350 learners today, but I'll show you our new building in a few slides, and that number is going to have to go up to accommodate the new research labs. Now, we have very large graduate medical education programs. These are the numbers in Worcester, but if you add the numbers in Bay State and in the Berkshires and at Leahy Clinic, be in the thousands of folks who are doing their residency training. So that's what happens after you graduate from medical school. You become a resident in a graduate medical education program. We're a very large medical school from a revenue standpoint, over $1 billion in revenues. Just to give you a benchmark, the Boston schools, BU and Tufts, are in the $100 million range, not more than that. And we're at $1 billion. We have more NIH funding on our campus than 26 entire states. And that's a testament to our outstanding faculty. And we've done a much better job in recent years in ranking our licensing revenue among U.S. universities. So I can tell you the 1 and 2 in the year that we were 3, the 1 and 2 were hospitals on the West Coast. So we were the first university in the nation. And you can pick whatever university you want. You name it, famous, not famous, begins with H, begins with P, begins with J, begins with M, you name it. We were ahead of them. Now, that's pretty exciting for us. We're always in the top 10 for primary care, although I have to be honest with you, I think the U.S. News and World Report reporting has got a little bit of trouble this year. It's been delayed now 4 or 5 times. And I'm very proud of the fact that our employees voted us one of the top places to work in Massachusetts. And we are the only educational institution in Massachusetts to be in the Boston Globe top places to work in 2022. So these are really wonderful accomplishments. Okay, next slide. Now, just to give you a sense, so this is the day when Dean Flock gets his grade for the year. He likes to say that. He says, if we have a great match, he's had a great year. And once again, we had a terrific match. We had 60s. So there's been some question, we're just a big research institution now. And do we have any fidelity left to our primary care mission? 66 of the 149 students went into primary care. 61 are going to stay in Massachusetts, and 22 are staying at our own institution. And I think that says volumes. If we add Bay State and Leahy, which we'll do in future years, that number is going to be into the 30s. And that's it. When our students want to stay at our place, that's terrific. Now, we are the Massachusetts medical school. Okay, call it what you want. We are all over the Commonwealth. Where the red dot is, that's Worcester. And that's where our 2 principal affiliates in Worcester are, UMass Memorial and St. Vincent's. But we now go from Cape Cod to the Berkshires, from Springfield to Burlington. And we've added a regional campus in Springfield. That's the picture on the left in 2015. We have 25 students admitted to the medical school who are involved in a program that focuses on urban and rural community health. And we just announced within the last few months, and great credit to the dean and his colleagues on this one, and both of these, frankly, in this whole notion of creating a branch campus. We are now going to have a program called Lead at Leahy. And the 25 students who are admitted to the Leahy track and will do all their clinical things at UMass Chan Leahy will focus on leadership, health system science, and interprofessional education. We're pretty excited about that. And I can tell you, if I were applying to medical school today, that would have been a program that I would want to be in. I think it'll be extremely, we had a great day up there a couple of weeks ago. We went up and met that there's a new president at the Leahy Clinic, Dr. Susan Moffett-Bruce. It was just really a spectacular day, just wonderful. Now, you are awesome supporters of our research enterprise. You all know this is Craig Mello's picture. He won the Nobel Prize in 2006. We're now, when Terry and I got to the medical school, there was about, $120, $130 million in research. We've almost tripled that in the 16 years we've been here. So that's really pretty exciting. These are the areas that we're focused in: neuroscience, RNA biology. Coming out of the Nobel Prize, we built the Sherman Center. There was a great focus in RNA biology. Infection and immunity, Dr. Harris will talk about that in a moment because he's now heading up an institute for us. He's a rare breed. He's a great doctor. He's an outstanding teacher, and he's also a very, very innovative and forward-thinking investigator. And he approached us to say, I think there's a lot of things we could do in an institute around immunology, and so we're very excited about doing that. Gene therapy and rare disease research, cancer, and I'm very, very bullish on our efforts now around health equity. We're spending a fair bit of time. I would say one of the big ads in this, the third version of the strategic plan, was to have a much greater focus on health equity. And in a future meeting, we might introduce you to a few of the folks who have come to lead that effort for it, because they are really spectacular. Now, this is a great picture. I was going to have Brendan show you the picture from our first year first, but this is a picture of the endowed chairs. And we're having a hard time now because the room is not big enough to get the whole crowd. We're now at 63 or 64. And we have a couple more coming in, and we still have needs like for John Harris to get an endowed chair, and we're working hard on that. I was on some visits recently thinking about that with some donors. So anyways, this is a really wonderful statement that this many faculty now have endowed chairs. And look at the number of women. There was one when we got here. There are 22 now. And now I'm not going to be happy till there's parity, but we're making great progress. Some very exciting news in the last few weeks. Kate Fitzgerald and Phil Zamore were elected to the American Academy of Arts and Sciences. And just this week, Dr. Zamore was elected to the National Academy of Science. So this is really amazing that we continue, so we have two faculty members into the American Academy of Arts and Sciences and another one into the National Academy of Science. It's really, and as many of you know, we lost a dear friend and colleague in Michael Green a few months ago. This was something that he spent a lot of time working. He was very supportive of getting our faculty members recognized by the Academy. So I'm very excited about this. Now, let's think about the future together just for a little bit. I'm constantly thinking about how we're going to invest in the future. So let's look at the campus quickly here. We're going to show you the next slide. So the building in orange is the new VA outpatient clinic that we created. This is what it looks like if you go up to it. This is, there's a long-standing relationship between American medical education and the VA, and I'm very proud now that we have a VA focused effort on our campus. And I can tell you the veterans are thrilled that they can come to our campus, and now if they're seen in this building, they can actually come across the way to the hospital to be seen further. So, and again, this was right out of the plan. We said we need to do more to expose our students and residents to VA patients to our campus, and here's our effort. If you look at this is the Lazar building. Many of you were here when that was built. We came just after it. If you look at the next building, this is the Sherman building. This is the building that we, I have to be honest with you, the board thought I should have my head examined because we said we were going to do this in 2008, 2009, in the midst of recession. I think it was the best thing we ever did. We built the building. We got great workmanship because there were no jobs. We came in $17 million under budget. We were going to shell the top two floors. We not only finished them, we occupied them, and still $17 million under budget. So it's amazing. And the biotech park, we used to have it, we lost it, and we reacquired it, and that's going to be like our venture development center into the future. The folks who are in Biotech 2 are going to go into this new building now, which is the new education and research building, and that's what it's going to look like. And as they come into Biotech 2, then from Biotech 2 into the new building, we will renovate Biotech 2 and then put that into the Biotech Park. But this is really, look at how beautiful that building is. It's going to frame the campus really very specially. And from sort of the top down, our gene therapy center, all things neurology and neurobiology will be next, the program in molecular medicine, and then the program in human genetics and evolutionary biology. So those will be the scientists. And this is sort of like a real world picture. That's we couldn't find a sunny day this week, but anyways, that's what it looks like. And it's all, I got a chance to go in there a couple of weeks ago, and it is, man, I'll tell you, it's going to be beautiful. It really is going to be beautiful. And when you get up in the top and you look out at the campus, man, it's really special. So we're just about to I mentioned this at the beginning. This will be how I conclude. Just about to have our 50th commencement, and we're very excited about that. And these are the folks who are going to come and we're going to honor. Katie Couric, I'm sure most of you know Katie Couric. She was a lead talent on the Today Show forever. Very sadly, her husband at a young age developed colon cancer and died. And subsequent to that, she has raised $750 million for research. And when I went to see her, I said, Ms. Couric, I know you're giving a lot of commencement addresses, but I'd like to give you the opportunity as a patient to speak to American caregivers. And we will give you every camera in America will be there. Why don't you come and talk to our folks, and by doing so, talk to America. Luke O'Neill is one of the world's foremost immunologists at Trinity in Dublin. Just a wonderfully gregarious guy. For those of you who come to any of the commencement addresses, he's just constantly has a smile on his face. He doesn't want me to say this, but he's more or less the Tony Fauci of Ireland. He's been the person who was kind of at the forefront of helping Ireland deal with all things COVID. And Tanya Saman, the Saman family and their foundation have provided us with 2 endowed chairs, a $3 million gift. And we're going to honor the family, particularly Tanya. Her husband, Maroon, called me a few years ago from Myanmar with a headache and had a very large brain tumor. And just a few days before he died, he asked to see me, and he asked me what I would like. And I said, if you would do 2 endowed chairs, one for our chair of neurosurgery and one for our leading scientist, that would be great. So we're gonna celebrate the Saman family. It'll be really a terrific day. So as you can see, it's really a very wonderful time on the campus. And let me end where I began, which is expressing my gratitude to all of you for your continued generosity and support. Indeed, it makes an enormous difference. Thank you very much.
Thank you, Chancellor. Thank you, Chancellor Collins. And I used the word momentum, and it's a word that you used at our convocation recently. And we remember from our physics class in high school, it's mass times velocity. The mass of this institution over 50 years is extraordinary. The velocity that you have described is extraordinary, but I think the equation is wrong. It's mass times velocity as the result of cerebral foreplay and strategic thinking. So I applaud you. Thank you. Now it's another pleasure and privilege for me to introduce Dr. Terry Flott, who is the Executive Vice Chancellor, Provost, and Dean of the UMass Chan Medical School, and also our Chief Scientific Officer. And I might just say, without giving him an elaborate introduction, which he surely deserves, I have the privilege of working closely with him as I am the Associate Vice Provost for Research, and I marvel at his leadership on that domain while running and developing a gene therapy center that the Chancellor alluded to, which is in the one particular branch of gene therapy using a particular viral vector, AAV, the tops in the world. And there is great progress in that field, both here at UMass under Dr. Flott's leadership, along with Dr. Guangping Gao, and in the world at large. So Terry, thank you for joining us, and I now turn it over to you to introduce our featured speaker, Dr. Harris. Thank you, Terry.
Great. Well, thank you, Tharu, and thank you for your kind words, also for your stewardship and advocacy and leadership of the Hudson Hoagland Society, and also your advocacy for our faculty who were mentioned moving forward in their academy memberships, which is another index of our great momentum. So it is really a thrill and a pleasure for me to have the honor to introduce Dr. John Harris to give this year's Hudson Hoagland Society keynote lecture. So as the chancellor mentioned, John is a double alum of UMass Chan Medical School. He's an alum actually of both the T.H. Chan School of Medicine and the Morningside Graduate School of Medical Sciences. He was an MD PhD student in our MD PhD program a number of years ago, and we're so proud of everything that he's accomplished. He's currently serving as the professor and chair of the Department of Dermatology. He also, as the chancellor mentioned, in becoming chair, also founded this Autoimmune Therapeutics Institute, was also the founding director of the Vitiligo Clinic and Research Center at UMass Chan Medical School, which is a wonderful, unique integration of basic science, translational science, and clinical care that really does bring hope and really has, Chancellor, already altered the course of history of vitiligo as a disease. So as I mentioned, John's got his MD and his PhD here at UMass Chan Medical School, actually in the program of molecular medicine. He actually worked with Dr. Aldo Rossini, who some of you might have remembered, working on type 1, that is autoimmune juvenile diabetes. And then he went on to UPenn in a combined research residency program in dermatology. And in his postdoc there, developed a highly important model, the mouse model of vitiligo, that actually reproduces the phenotype of depigmentation, which has become, which was the initial tool he used for a lot of his early work and testing interventions in that condition. But as he'll talk about, he's gone far beyond mouse models, but still iteratively uses those models. He is a consummate physician scientist, educator, as Chancellor Collins mentioned, advising numerous graduate students, medical students, MD, PhD students, postdocs, and proliferating his knowledge and works through publications of great impact and patents and presentations and really staying in touch with both the most basic science and with the patient communities. He's one of our most active Twitter users among our department chairs, I have to say, and uses that to great effect to disseminate knowledge and interest in the disease. I won't spend a lot more time talking about John. You'll be incredibly impressed if you don't already know him, but I do want to comment on his terrific title for his talk, Defeating the Enemy Within. It called to mind for me, I'm going to channel Thoreau here a little bit and pulling out an old reference. It called to mind to me a term that I think was coined by Paul Ehrlich, one of the classic immunologists in the founding part of that era, where he talked about the phenomenon of horror autotoxicus, which is the phenomenon describing the fact that as things normally sit, the immune system, which has great power to destroy, destroys the pathogens and leaves the host alone. But when that goes awry, then those weapons are turned inward. And so he'll talk a little bit about that and about how a breakthrough in skin disease advanced the treatment options for autoimmunity more generally. So John, I won't take up any more of your time. We're waiting very much in anticipation for your talk. So thank you.
Thank you, Dean, for that wonderful introduction. I can't take full responsibility for that title. This was a collaborative effort with Kim and Thoreau, and to really put together an idea of what we're doing and what our goals are. I'm going to stop sharing here so I can share my own screen. And I have to say, this is such an exciting place to be. I've been here, as you mentioned, as an MD PhD student, and then as an intern, and then back as faculty. And UMass is an incredibly special place. And one of the reasons why, and it's recognized all over, is how collaborative we are. So we work as a team, we live as a team, we succeed as a team. And that's a major theme of my talk today is collaborations here, and how much we've accomplished with others as teams. I also, like everyone else, want to thank those that are out there and everyone who has supported research here at UMass Chan. We have amazing scientists here, as you know. And philanthropic support is really a key in promoting our work and giving us freedom to be able to explore new ideas. A lot of what I'm talking about today were new ideas and unfunded ideas that were only funded by philanthropists and foundations. And some of the philanthropists that funded this work are on the call today, and I want to thank you because it's really made a huge impact. Let's see, I'm going to show my disclosures here. There's a lot of companies on here. I work with a lot of companies to help them develop drugs for vitiligo. It's a super exciting time. Over the next 10 or 20 years, it's going to be pretty incredible what we're able to offer patients. I want to start with this story about Aldo. I'm going to hide the floating meeting controls. I learned how to do that from Dean Flott today, how to hide the meeting controls so I can see my own screen. So Aldo was, as mentioned, was my MD PhD advisor. And I worked in his lab and studied type 1 diabetes and fell in love with the concept of autoimmunity and how to defeat it. But I can say after a couple of years, I was incredibly frustrated with the fact that we were studying animal models and cells in a dish, and we weren't actually studying patients. And the reason, of course, in type 1 diabetes is the pancreas is very difficult to get at, to get sample tissue. And so I told him, I'm not sure what I want to do, but I want to study people. And he said, you might want to consider dermatology. And it's something I'd never considered, but that was the beginning of a seed planted. But really, the major event in my life happened when I ran into Aldo late one night in the hospital. I was on call as a student after finishing my PhD. It was about 10 o'clock at night. He was walking the other way down the hall and we ran into each other, and he said, John, and he said, I have a patient I want you to meet. And he said, follow me. So I said, okay. I followed him to the intensive care unit to meet SP, who is a 21-year-old woman who was admitted for new onset type 1 diabetes. She was in diabetic ketoacidosis, but doing actually really well with treatment as expected. And so after I met her, he said, I want you to do a physical exam. So I listened to her heart and it was fine. And I sat her up to listen to her lungs, and I noticed a large white spot on her back. And I immediately recognized that as vitiligo, an autoimmune skin disease. And so I said, well, when did that happen? And she said, oh, it all happened at the same time. On further exam, I found an enlarged thyroid and found out she was later diagnosed with Hashimoto's thyroiditis and pernicious anemia. And I recognized right away that she had a coincident onset of 4 autoimmune diseases: type 1 diabetes, vitiligo, thyroiditis, and pernicious anemia. And it was then and there I said, I want to be a dermatologist. I want to be a vitiligo expert. I want to study vitiligo and understand how it works so I can get insight into how autoimmunity works in people. And at the time, vitiligo was a human model of type 1 diabetes, so that I could someday circle back and try to develop a cure for diabetes. Now, of course, I'm in love with my patients and my work in vitiligo for its own sake. But again, one of the themes of this talk is going to be how we think we'll be able to translate our work beyond just vitiligo. So vitiligo is a common autoimmune disease. It affects about 1.4% of the population. That's a lot of people. And about half the patients who get it before the age of 20. So children get it, they have it for the rest of their lives. As a physician, I'm managing in my clinic, managing patients over decades of life and optimizing their treatments that way. And as a scientist, it gives me an opportunity to study their disease over decades of life as well. And we've been able to learn a lot so far, and there's still so much to learn. As I mentioned, vitiligo patients and their family members have strong associations with other autoimmune diseases, including type 1 diabetes. And I just included these photos from a couple of patients I've seen recently in my clinic. You can see the vitiligo here. This patient's actually responding very well. All these little brown spots are the color coming back as we treated him. This is a glucose sensor because he has type 1 diabetes. This is another patient with vitiligo and an insulin pump and actually an ostomy because she has Crohn's disease as well. And so there's clearly an autoimmune predisposition for vitiligo in their family members. So what is autoimmunity and how do we fix it? It's an important question. And autoimmunity is essentially the immune system producing a number of damaging factors that can damage a number of different organs. And vitiligo is in the skin. You can get lupus in the kidney and the lungs and MS in the brain and lots of things. What influences the immune system include our genes, our genetic inheritance, as well as our environment, environmental exposures, and other things. And so one of the questions is, can we shut this down? Can we shut this off, right? And we know that's the case. We have drugs that will turn off the immune system. And we actually use those for organ transplants to prevent rejection. That's important. And I had this patient who came into my clinic one day with widespread vitiligo. You can see the depigmentation here. And he had received a kidney transplant for unrelated reasons, and he was on kidney transplant level immunosuppression. I noticed that he repigmented faster than anyone I'd ever seen, and telling us that if we put all of our vitiligo patients on kidney transplant level immunosuppression, their skin would do great. The problem is the rest of them would not. And so when we immunosuppress people and turn off their immune system in a large swath, infection is a major problem. And this can be really highly morbid and even cause mortality. So there's a concept that we can develop more targeted immunosuppression or more targeted drugs to turn off parts of the immune system. We can turn off antibodies now, we can deplete T cells, we can get rid of large numbers of cytokines. But what's even better is to try to develop more targeted, developing drugs that target just parts of the immune system, parts of the T cells, subsets of T cells, maybe just one cytokine that's responsible for driving disease. And as you can imagine, leaving the rest of the immune system intact to protect us from infection. And so that was the concept here that came to my mind when I saw this patient. I thought, can we develop a targeted immunotherapy to treat vitiligo that'll improve treatment responses and still be safe? And we know we can do this in psoriasis and eczema. We can target a single cytokine and turn off the disease for most patients, and that still is safe. But the problem is those drugs don't work for vitiligo. And so we set out to try to find a pathway that we could target in vitiligo to accomplish the same thing. And so, as Dean Flott mentioned, our strategy is to integrate basic science. This is a mouse model of vitiligo that I developed during my training. These are white spots on the skin. This is from vitiligo, and they get it on the feet, the nose, and the ears. And so we study basic mechanisms of disease in the mouse model, but we also study tissues from our patients from clinic. And this is translational research as we study patient tissues to understand what's going on. And then we also conduct clinical trials to test new treatments for patients. And we try to integrate this all seamlessly to develop a mechanistic understanding of disease that's clinically relevant to develop new therapies. So the first story I want to tell you today is a bench to bedside story, and it begins in the mouse model. And it actually begins with Mehdi Rashigi, who was a postdoc in my lab at the time and now has his own lab here at UMass Chan and studying other autoimmune diseases. But what we identified in the mouse was that vitiligo was really driven by a cytokine called interferon gamma. And that cytokine turns on other downstream chemokines, including CXCL10, and we identified this in our patients using human tissues. And interferon gamma, in order to turn on CXCL10, it signals through a series of proteins, a family of proteins called JAK kinases, Janus kinases or JAKs. And so we had this idea that if we could have an inhibitor of Janus kinase, we could block that interferon gamma signaling, and that would stop the disease. And so we actually had an opportunity to treat one vitiligo patient with oral ruxolitinib, a JAK kinase inhibitor, orally, and he repigmented very, very well, very quickly. So you can see most of the pigment coming back on his face. And so this really provided the rationale to conduct a number of clinical trials that are ongoing in vitiligo to test JAK inhibitors. The very first one was putting ruxolitinib into a cream and testing that. And the phase 2 clinical trial was very effective. The phase 3 clinical trial was very effective, and that resulted in FDA approval of of topical ruxolitinib for vitiligo. This was the first FDA-approved treatment for vitiligo. It's now called Opzelura, and you may have, if you haven't yet, you'll see commercials on TV for Opzelura to treat vitiligo. It's a very exciting time for our patients. I can tell you after FDA approval last summer, I got about 300 phone calls from my patients within a couple weeks, and we were knee deep in prior authorizations. But we've been able to get this for our patients, and it's working, and they're excited. So the second story I want to tell you is a bedside to bench and back, and it starts in the clinic with the observation that patients get vitiligo in a certain area of their skin, and we can treat them with lots of treatments and make them better. But when we stop the treatments, it relapses. And not only does it come back, but it comes back in the exact same place it was before. So there appears to be some memory of the skin, remembering it's supposed to have vitiligo, so that when the treatment is stopped, that brings it back immediately. And this largely is Jillian's work. Jillian Richmond was a postdoc in the lab. She now has her own lab here at UMass Chan studying other autoimmune diseases as well. And so this observation in the clinic made us go back to the bench to try to understand what the source of this memory in the skin was. And we discovered a T cell that forms in the skin during vitiligo called resident memory T cells. And we identified them in the mice. And then we also looked in our vitiligo patients, and we've identified them there as well. And then importantly, we learned and discovered that these resident memory T cells, autoreactive cells, are absolutely dependent on IL-15. This is a cytokine that's produced in the skin, and they need IL-15 signaling to stay there in the skin and cause disease. So when we blocked IL-15 signaling, we saw our mice repigment, similar to the JAK inhibitors actually. But in this, now we could stop the therapy and they continued to repigment and had durable responses without relapse. So that was pretty exciting. That provided the rationale for new clinical trials. I was able to get funding through the Immune Tolerance Network at the NIH to test an IL-15 antibody in vitiligo patients. This is an ongoing clinical trial now. I was the PI of that trial. I wrote the protocol for that trial, but had to recuse myself when we received venture capital investment to start a company called Vitiligo Therapeutics. And we developed an antibody against the IL-15 receptor to turn off IL-15 signaling. For a number of reasons, we think IL-15 receptor is a much better target therapeutically. And over the past 4 years, we developed a drug called auremolimab. And we're very excited that in December, our company was acquired by Incyte, and we're working with them now to initiate clinical trials very soon, within the next few months, hopefully this summer. So another opportunity, a second story to develop a new therapeutic for vitiligo. So this is a quick summary of that piece. CD8 T cells come into the skin, they find melanocytes, they start making interferon gamma, which turns on chemokines that recruit more T cells into the skin. And this constant positive feedback recruitment drives the progression of vitiligo. And I often say this is very similar to the way ants discover food. So if you're in your driveway this summer and you're eating an ice cream cone and you drop some ice cream on your driveway, and an ant finally stumbles on that ice cream, it'll lay a trail back to the nest, and within just a few minutes, you'll have thousands of ants on that ice cream. So that's similar. This is the first ant encountering and laying the trail and recruiting more cells. And while they're in there, a subset of these cells create what's called resident memory T cells that glue themselves into the epidermis and stay there forever. So when melanocytes try to crawl back into the skin and repigment the skin, they engage with them, make interferon gamma, and reinitiate the whole process to cause relapse, to cause maintenance of the disease. If we use treatments to turn them off and put them to sleep, this allows repigmentation of the skin. Patients get better, including JAK inhibitors, but as soon as these are stopped, the cells are still there, they wake back up and reinitiate the disease and relapse. And so if we block IL-15, we can erase this memory and allow durable long-term treatment responses. Okay, so there are two pathways that we are working on to develop new therapies, including JAK inhibitors, interferon gamma signaling, as well as IL-15 inhibitors. But we were convinced, based on other diseases and our knowledge of the disease, that there must be other pathways that we could target therapeutically, and we may be able to develop much better treatments as well. So we wanted to dig deeper and discover other pathways to target other than those two. And in collaboration with Manuel Garber, computational biologist here at UMass and close friend and collaborator here, we decided to acquire cells directly out of the skin of vitiligo patients and analyze them using really cutting-edge technologies. And to do this, we developed skin blistering. So we create a little suction on the skin, and after about 30 minutes, a blister forms with fluid in it. We can do this on affected vitiligo skin, normal skin from those patients, and healthy skin. And we can take the cells out of the skin and analyze them here with single cell technologies. We can take the fluid and analyze them using proteomics and look at metabolites, et cetera, to really dissect all of the amazing cell-cell conversations that are happening in the skin that drive vitiligo. Here in the proteomics, you can see CXCL9 and 10. Remember, that's interferon gamma-induced chemokines that drive the disease. So we could see that it's confirming our previous data. And based on the data that we've generated together with using those technologies, we were able to get an NIH P50 award that started a little less than a year ago to create a Vitiligo Center of Research Translation. This is a $5 million award over the next 5 years to dig deeper into that data that we had generated and really better understand in context how cells communicate with each other to drive the disease. And so as we better understand these pathways, we can develop treatments to target them that may work even better than what we've discovered so far. And this is a collaboration of a team, as we always like to work. This is Manuel Garber again on this grant, and then Jillian Richmond, who's now independent, contributing the cytokine work, and Sally Kent from the diabetes group, and Larry Stern from pathology and immunology to really help us to dissect all of these pathways in an incredible way. To understand disease. We're also collaborating with Long Kai at Caltech, who has a spatial transcriptomics center. He's a world leader in this opportunity to investigate how cells are positioned in the skin and how they interact together. So lots to come. We just finished our first year, and we've already generated lots of data, and we're excited for the future. We also use this data to get an NIH U01 award that just started last week. And this is a large award. This is a $4 million award over 5 years to study what causes autoimmunity. And we're using vitiligo as a model to do this. And this is also a team effort. Again, Manuel Garber, this time Mehdi Rashigi is helping us on this project. We have a collaborator from MIT, Daryl Irvine, who makes absorptive microneedle patches, and Richard Spritz from the University of Colorado. And in short, we're going to recruit 200 vitiligo patients and 800 of their close relatives, because as you know from the earlier slides, they're at a high risk for developing vitiligo. We're going to follow them for years and try to predict based on their genetics, as well as some biomarkers that we find in their skin and their blood, who might be at highest risk for developing vitiligo. And this project can teach us how to potentially prevent autoimmunity. And again, because of the shared mechanisms with other autoimmune diseases, we think this will translate to others as well. So we're using a Tasso device that we can mail to patients. They can draw their own blood and mail it back. We're using tape skin strips where we can collect just the surface of the skin and learn lots about what's happening inside, as well as absorptive microneedles that will actually absorb a little bit of skin fluid and allow us to analyze what's going on there. So very excited about that. There's a lot of work to do, but we expect big things. All right, so in this next portion of the talk, I want to talk about how we're taking that data and really working with others and collaborators to develop novel treatments for vitiligo. And I call this right place at the right time for two reasons. Because first of all, we're trying to get the drug in the right place at the right time to avoid side effects. But we're also, the reason we were able to develop this is I was in the right place at the right time to work with collaborators who really transformed our work. So you probably all know, that Craig Mello won the Nobel Prize for discovering RNA interference in 2006. I happened to have just started my dermatology residency at Penn at the time, so I was not here. I felt like my whole family was celebrating an important milestone, and I missed it, but I celebrated from afar. So RNA interference can be, in theory, used as a drug to turn off proteins that drive inflammation or drive disease and are responsible for disease. And this was the hope and the concept very early on, that if you take this double-stranded RNA and you know the sequence of whatever gene you're trying to turn off, you can have this enter the cell, chew up mRNA, which then prevents the protein from ever getting translated and produced. And therefore, the protein's not available in the cell to do the bad thing that it was doing, whatever that is. In our case, inflammation. But the problem is this as a drug really performed pretty poorly early on until Anastasia Kvorova, who is one of our amazing faculty members and a chemist, was able to convert it into a molecule that improved its potency, increased its durability, and allowed selective delivery to where it needed to be in vivo in the body. And so based on that, it now made the molecule possible to use therapeutically for human disease. And you heard last year about some of this, and it's just a super exciting time. There are now FDA-approved drugs that are siRNA drugs, and this was selected as, her discovery was selected as one of Nature Biotech's 25 landmark papers in the last 25 years. So this was a great opportunity. I met Anastasia in the hall and I said, hey, Anastasia, we got to figure out how to get siRNA in the skin to treat skin disease. And she said, oh my goodness, that should be easy. We can do brain. So let's start working right away. And we were able to recruit Chi Tang, who's a chemist, to be our postdoc, shared postdoc, who worked on this to develop an siRNA to treat skin disease. So our first target was to develop an siRNA against JAK1. Remember, the JAK inhibitors treated vitiligo. That was well validated, but JAK inhibitors are pretty dangerous systemically for side effects and have black box warnings from the FDA. So we wanted to develop, use siRNA to develop a selective JAK1 inhibitor. And he was able to accomplish that. So he was able to find a sequence to make a JAK1 siRNA that targets both human and mouse JAK1. And here in the red line, you're just looking at the disappearance of the mRNA as we introduce the siRNA into the cells. In addition, of course, the protein's important. And when the mRNA is gone, the protein's gone, and we're able to turn off JAK1 in these cells using ALD-102, which is the drug that he developed. So the next step was to see how selective is it. There's a whole family of JAKs, JAK1, JAK2, JAK3, and interestingly, TYK2. And this is exquisitely specific only for JAK1. We're able to turn off JAK1 without touching any of the others. And no small molecules can do that, including the FDA-approved ruxolitinib for vitiligo. So we thought we could test our siRNA against ruxolitinib head-to-head, because interferon gamma induces these chemokines that we mentioned before, CXCL 9, 10, and 11, through JAKs. And so we could actually treat cells with these two drugs and treat with interferon gamma and look to see what the effect was downstream. And essentially, we can see that interferon gamma turns these on here, here, and here. And that siRNA completely turns them off, as you can see here, here, and here, and actually to a greater extent than the FDA-approved ruxolitinib drug for vitiligo. And so we think that we're onto something. This is a really potent inhibitor. And so he tested it in our mouse model. He injected one foot pad with the drug and it stayed black, and the other foot pad with a control, and it did not. You can see the vitiligo forming here. You can see that it blocked interferon gamma signaling in the foot because CXCL 9 and 10 were reduced. And then the autoreactive T cell entry into the skin was also reduced significantly by this drug, really showing promise. So he then wanted to show that it worked in humans as well. Remember, the drug works both in mouse and human. So he labeled it with a fluorescent red dye and injected it into surgically discarded human skin. And we can see where it goes, diffuses out, goes throughout the skin. In all layers, it's present. It knocks down JAK1 in human skin, as you can see here, and it inhibits interferon gamma signaling, as we mentioned before, looking at the chemokines, CXCL 9, 10, and 11. So it's functionally effective in human skin. So we're excited about this opportunity. We actually started a company called Autoimmune Therapeutics. I'll tell you a little bit more about that. And we're excited about taking that into clinical trials. So the second story I want to tell you is titled Developing a Skin-Tethered Bispecific Antibody to Treat Vitiligo. And here, the right place at the right time was being at Penn and training in dermatology, where our chair of dermatology, John Stanley, studied a disease called pemphigus vulgaris. This was his research interest. He had a clinic seeing these patients. I worked with him in this clinic. And essentially, pemphigus is caused by an autoantibody that patients develop that targets their own skin and causes it to be unglued and fall off and create blisters. And this is a deadly disease. But in his process of studying this disease, he found that some of these patients would make antibodies that bind to their skin but don't cause blisters. You know, while some antibodies cause blisters, others just bound to the skin and sat there and didn't do anything. So he thought, could we take this horrible disease, isolate an antibody that targets the skin and binds the skin, and use that to deliver drugs therapeutically so that it only goes to the skin and doesn't go anywhere else? So Chow was a grad student in the lab at the time who was an antibody engineer, and he loved this idea. And he said, I want this to be my project. So he created a bispecific antibody, started with an antibody that blocks interferon gamma in red here, and the other antibody from the pemphigus patient that binds to the skin, desmoglein 3 protein in the skin, and then put those two together to create a bispecific antibody that binds to the skin and blocks interferon gamma. So he tested its ability to block interferon gamma. He showed that it was just as good as the interferon gamma blocking antibody here, whereas the skin binding antibody, of course, did not. And then he also tested its ability to bind to the skin, keratinocytes in the skin, and showed that it bound to the skin just as well as the original antibody, whereas, of course, the interferon gamma antibody did not. He then labeled it with radioactive iodine and injected it into 1 foot pad of a mouse, in addition to the untethered interferon gamma antibody and then the bispecific antibody. And you can see the untethered antibody comes out of the foot pad into the systemic mouse and diffuses all over. And this causes immunosuppression in this mouse. Whereas the bispecific antibody stayed in the foot pad. A little bit came out at 4 hours and then it disappeared and remained there long term. So we're excited about that. Here's a video of a whole mouse, and you can see that the untethered antibody going everywhere and the tethered antibody remaining in the injection site, and it's in the heart, but in the bispecific is not. So then he tested it in our mouse model, and here injected just this foot pad, and you can see protected just that foot pad, whereas the other foot pad got vitiligo, protecting here and not protecting here. Whereas the interferon gamma antibody that's untethered went from 1 foot pad all over the mouse to protect both. And then the skin binding antibody didn't protect either one. And so this really told us we were able to develop an antibody that would remain local, not cause immunosuppression elsewhere, and treat disease. So excited about that opportunity as well. So here's a summary of this. We showed you blocking JAK, using JAK inhibitors, blocking IL-15 are valuable. We can use conventional antibodies, bispecific antibodies, next generation siRNAs to do this. And we're working on moving these forward to treat vitiligo. And so for the last proportion, just for the last few minutes, I want to talk about how we can make this work for other autoimmune diseases. Remember, we started with type 1 diabetes. We went to vitiligo with the idea that someday we could maybe return. And this George Eisenbarth was a type 1 diabetes researcher who won the Banting Medal in 2010 and gave a lecture where he actually said type 1 diabetes is vitiligo of the pancreas. And the reason was because the infiltration in the pancreas is patchy, similar to the patchiness of vitiligo. And Aldo actually told me this, that he had been at the Banting lecture, and he came back and said, John, you wouldn't believe it. George Eisenbarth said that type 1 diabetes is vitiligo of the pancreas. And if you look, it's actually not that surprising. So Richard Spritz did lots of genetics, and these are the genes implicated in vitiligo along the top. And these are lots of other diseases and the genes implicated there, and you can see tons and tons of overlap of these genes. In particular, you can see type 1 diabetes sharing lots of risk genes with vitiligo. And so this probably explains why vitiligo patients and their family members get other autoimmune diseases, and also really strongly supports the idea that shared mechanisms drive all autoimmunity. And so if we can develop a treatment for one, they may work for others. And so this really formed the foundation, Aldo's real push for this idea that autoimmune diseases across organ systems and share mechanisms, and George's comment. We hypothesized that multiple autoimmune diseases can be targeted with drugs that treat vitiligo, and that we can use vitiligo, which is easily accessible in the skin, to study and learn about diseases in order to get a jump start on other diseases. So how can we leverage the translational data from the skin that we've generated to treat other diseases as well? And so we are working on novel drug delivery methods within, and this was the foundation for the Autoimmune Therapeutics Institute here that we formed, to really bring lots of autoimmune researchers under the same umbrella, physician scientists who see the patients, who do the studies, collaborate with basic scientists to develop new treatments and create this institute. So within the Institute, we're also developing new drug delivery methods. And so we're collaborating with lots of people, collaborating with Alison Keeler here, who is an AAV vector expert. We're a leading institution in AAV vectors, largely because of Dean Flott. And we're working together. We received a Riccio seed grant here at UMass Chan to study vectors to deliver shRNAs to treat vitiligo and other diseases. We also are working with Rafi Aroyan, who studies parasites of the gut. And we actually discovered together a parasite, one of Rafi's parasites. We can infect the mice and actually prevent vitiligo. We can't give them vitiligo once the parasite is in there. And so we're really now trying to understand, what is that parasite doing? Can we find products from that parasite that could be actually used as therapeutics? So we're pretty excited about that. You've already heard about the siRNA collaboration with Anastasia Kvorova to treat skin disease. We also collaborated with Kate Fitzgerald and Anastasia together to develop an siRNA that actually promotes melanoma immunotherapy. This was funded through a cancer seed grant here at UMass Chan. We formed a company called Autoimmune Therapeutics with a siRNA technology, as well as a technology from Georgia Tech, to deliver siRNA through the skin topically using these small star particles that can go into a gel and be rubbed on the skin and poke holes and allow penetration of the siRNA. So Aldine is funded through a $30 million Series A startup, and we're making great progress in showing that it really works. And then, of course, bispecific antibodies, as I mentioned. So the concept of the Institute is to take all of these treatment modalities that we've learned about and started to use, parasite products, small molecules, RNA interference, antibodies, gene therapy, and take all the delivery methods we've been working on, including orals and star particles topically and tissue tethering and AAV vectors to treat lots and lots of autoimmune diseases and other organs. So, for example, we think we could start with type 1 diabetes, say, hey, we're going to use a tissue tethering technique to deliver RNA interference. And this is one strong option that we're working on. We can also take multiple sclerosis and say, well, let's orally deliver parasite products to treat that disease. And that's another potential opportunity here. So you can imagine all of the potential combinations to treat disease. And this is why we think the Autoimmune Therapeutics Institute will be so powerful. This is the pipeline of drugs that we've already developed. Here are the JAK inhibitors that are now FDA approved. Here is auremolimab, developed by Valeris and acquired. Aldina has licensed the siRNAs. We're working on Helminth products, so lots of opportunities coming. So the major challenge in going from an academic target to therapeutic shown to work in humans is this valley of death. And this is a large cost to translate from understanding the target to actually developing the drug that can then be used in the clinic. And so we think that the AITI, this institute, will bridge this gap because we've done it before. So we've attracted over $10 million of investment into our academic work through, again, philanthropy, NIH funding, and others. We've attracted over $100 million in venture capital to bridge this gap to start companies, including Aldina and Valeris. And of course, Valeris developed auremolimab that was acquired, and so we successfully exited that drug. So we feel like we've created a path that we can follow for future opportunities. And to do that, we're recruiting physician scientists to work within the Institute. I'm there. Sarah Whitley is a recent recruit who studies hidradenitis suppurativa and already taking advantage of some of the opportunities that we've created. Mehdi Rishigi studied with me originally and now has his own lab studying lupus and dermatomyositis, awful diseases as well. And we're recruiting more, potentially physician scientists even that study other autoimmune diseases so that we can synergize and leverage our work to help them. Our collaborators are a huge population and really key to our work. And this is what makes UMass so special. So I'm going to end there and show you the group that actually does the work. These guys, I get to take a lot of credit for their work and their ideas, but really it's a team effort. It's a happy group. Lab meeting is my favorite part of the whole week because they're making really key discoveries and we get to help channel that. Of course, our funders, the NIH, through a number of grants, including the new P50 in U01, the Derm Foundation, American Skin Association. Medici has funded lots of the venture capital funding that we talked about. The Hartford Foundation, an anonymous donor, has supported our work and really enabled us to do lots and lots of these studies. Individual supporters out there also contributing. The Apple Pickers Foundation actually funded a crossover between vitiligo and type 1 diabetes and our collaborators. So I mentioned Twitter, follow me on Twitter and check out our website. And I want to thank you for your attention and your time.
Thank you, John. Wow. I want to mention that I had John in a class when he was in our PhD program, and he did well. And I can't tell you how proud I am, John, to see what you've done. As the dean said, as a physician scientist, and the extraordinary collaborative group, you don't have to reach extramurally in much of this, as you said, although there are some key people around the world. We have some questions. I'm looking at the chat box, and I think the first one is actually from Dean Flott. What is the biological explanation for why some autoimmunity is more commonly manifested in women? And this would be like lupus, for example, and can this mechanism be a clue to therapy?
Right. Yeah. It's an important question. Some autoimmune diseases are much more common in women, to the level of like 9-to-1 in lupus. And dermatomyositis is more common in women, and thyroid, Hashimoto's thyroiditis is more common in women. Vitiligo is not. Type 1 diabetes is not. And there have been a number of hypotheses. One is that there are inflammatory components that are encoded on the X chromosome. And so one idea is that if you've got two X chromosomes, sometimes the one that's supposed to be off is turned on, and you get a double dose of an inflammatory gene, and that actually makes you more susceptible to inflammation. Another explanation that actually has a lot of data behind it now, a friend of mine actually at University of Michigan, a dermatologist, really asked this question, and he found that skin from men and skin from women, actually, if you do RNA sequencing, you can see different levels of a number of different genes. And some of them are transcription factors that turn on inflammation. And so VGLL, I think, is one, and that is actually more highly expressed in women's skin. And that actually induces type 1 interferons that then drive lupus. And so it's not that being a woman gives you vitiligo, right? But it having that, you're a little bit more on edge in terms of your inflammation. And that's more easily tipped into disease. And so there's a number of hypotheses, but I think those are good ones.
Great.
And therapeutically, sure, if you can shut off, if you can correct that overexpression of that, transcription factor, whether you're using HDAC inhibitors and things to turn on and off, global genes, that might work.
We do have a couple of questions that came in via email, so I'll ask a couple of them now. Is there a connection between the microbiome and autoimmunity?
Yeah, so probably. The microbiome is super complex, and we are very interested in that. So we're actually working with the NIH now. They have an initiative for anybody who's listening that does research. The NIH has a new interest in the exposome, which is essentially anything you're exposed to externally that can influence disease. The microbiome is one of the very important pieces of the exposome, and we're applying for a grant actually to incorporate microbiome research into our U01. There's one mouse study that suggested that the microbiome influenced vitiligo in a mouse model. And then there's an important study in melanoma immunotherapy. So we've got, in the past few years, we've been able to cut the brakes of T cells in melanoma and allow clearance of the tumor. But it works in some and not in others. And actually, there was a strong paper that showed that the microbiome could determine who would respond to melanoma immunotherapy and who didn't. And so that's really important, in melanoma immunotherapy. But melanoma immunotherapy is vitiligo. Essentially, you're causing vitiligo in melanoma patients in order to clear their tumor. So because the microbiome was so important there, we strongly believe it's going to play an important role in vitiligo as well.
And another kind of connected question: what is the role of genetics in all of this?
So if you have vitiligo, your first degree relatives have a 6-fold increased risk of developing vitiligo. So the risk of vitiligo is about 1%. And if you've got a family member, it's 6%. So that's a big increased risk. That tells you that genes play an important role, right? But it's not 100%. And interestingly, if you have two identical twins, the risk, if one gets it, the other one gets it is about 23%. So much higher because they share all their DNA, but it's not 100%. So it's not all genes. And so people often say, well, it must be the environment. There must be genes plus your environment, except that identical twins probably share the same environment most of the time. And so there's also a role for chance, we think, that also influences whether you get disease, even with the same genetic deck. So if you have a deck of cards, and you have two decks that are exactly the same, you can deal a different hand. And we think that's how the immune system operates, and that might be why genetics plays a very important role, but not the whole role.
Two more questions. One relates to the patient that you spoke about at the beginning of your talk, who had multiple autoimmune diseases seemingly at the same time. Were you ever able to determine which came first? And if so, does that impact the treatment that they then receive?
I was very interested in that question, and I asked her right away. As soon as I discovered the vitiligo, I said, "When did this happen?" And she said it all happened at the same time, indicating that she just got barraged with 4 autoimmune diseases all at once. In my clinic, I see lots of patients with vitiligo and other diseases, and there's no strict rule which one comes first. So sometimes they get vitiligo first, sometimes they get it second. And we think it's probably because their genetic makeup is really prone to autoimmunity. And so it's just, again, putting them on the edge, and when they tip for each disease could be different and have different effects. There are actually environmental factors that induce vitiligo. Believe it or not, hair dyes have been shown to increase the risk of developing vitiligo by about 50%. There was an outbreak of vitiligo in Japan in 2013 because there was a new skin lightening cream that everybody started using. Over 18,000 people got vitiligo from it. And so these chemical exposures can also induce disease. And so it's very complex, kind of the interplay between genetics and chemicals and when these happen. So you can imagine somebody predisposed might get vitiligo first if they're exposed to certain chemicals, and might get it later if they're not, or might not get it at all.
And this viewer wants to know, how do you stay up to date with all the latest developments and breakthroughs in the field of autoimmunity with all that you have going on? How do you stay ahead of everything?
It's like, if there's any sports fans in the audience, if you love baseball, how do you stay up to date with baseball? Well, you watch it, right? And you talk about it. And I think that's part of it. Autoimmunity and research is so fantastically exciting. And new discoveries it's not a chore. And then also on top of that, we also often will say that our trainees really teach us. So they're finding papers, they're getting ideas, and they're teaching us those ideas. And that's part of the process. And going to meetings, I'm going to Tokyo next week to a great every 5-year skin disease meeting. We'll meet a bunch of friends there, see a lot about autoimmunity in the skin, share a lot of our own data. And so it's just that's part of the process and the love of what we do.
Terrific. I think that's all the questions, Thoru.
Well, John, I want to just make one final point, and that is the entrepreneurial axis of all you're doing. I think the audience needs to know that the university believes, as a public institution in the Commonwealth of Massachusetts, that we have an obligation to transfer discoveries into the corporate sector under the proper policies and controls. And I would just say that I have the honor to again be involved with John, because I am the research integrity officer of UMass Medical School. So no longer is he in my class, but I now have the privilege of looking over these commercialization activities, which are all being done under the proper policies of the university and with a great deal of transparency on John's part. Thank you again for all you're doing on the frontier of autoimmunity, and particularly the possibility that the vitiligo axis could be an opening window to other autoimmune diseases.
I don't think I could have accomplished this anywhere else. I think UMass is such a special place and really enables all of this. It's really innovative. It's exciting. It's growing, and we're smaller and we can pivot and we can pursue really exciting things. So it's awesome. Thanks, Thoru.
I think you just broke up, but maybe that's a great segue out to say that also ties to the chancellor and the dean's leadership to make UMass the incubator that it is, which you are dealing with so effectively and so productively. So thank you all for your support. Thank you, John, again, and everybody have a great evening. And thank you for being members of the Hudson Hoagland Society. Good night.
Good night.
Good night.